Wild birds survive intense seasonal volatility by executing drastic physiological shifts throughout the year. They rebuild their muscle mass, grow entire new coats of feathers, and grow or shrink their reproductive organs to match the calendar. This creates a strict evolutionary trade-off. A species must choose between rigid scheduling to survive highly predictable seasonal extremes or overlapping physiological states to exploit unpredictability. Panel A shows the Gambel's white-crowned sparrow, isolating migration, breeding, and molt. This maximizes tolerance to regular seasons, but sacrifices agility. Contrast this with Panel D, the zebrafinch. Overlapping rings maintain continuous breeding readiness and molt to survive unpredictable arid environments. A bird's survival strategy is defined by whether it isolates or overlaps its life stages, setting the parameters for how it interacts with its environment. By what mechanism does a bird translate environmental changes into these rigid physical transitions? Short-term environmental cues, like an unusually warm week or a sudden abundance of seeds, are too erratic. Relying on them to initiate a massive physical overhaul like molting would leave a bird vulnerable if the weather suddenly reversed. To make a long-term physiological commitment, birds rely on photoperiod, the length of the day. It is the only astronomically reliable metric of seasonal progression. To understand avian phenology and the pressures of rapid climate change, we must deconstruct the hard-coded neuroendocrine architecture driving these schedules. Avian timing is governed by a strict neurological cascade. Experiencing long daylight hours is mechanically insufficient on its own. For the biological trigger to flip, that external light must intersect with a highly specific internal lindro, a process known as coincidence detection. This graph maps a bird's internal 24-hour circadian clock. Notice the sharp peak between hours 12 and 20. This is the photoinducible phase. The bird's reproductive system is only sensitive to light during this exact window. During a short winter day, the sunlight cuts off before this internal window opens. Because the light and the inducible phase fail to coincide, the reproductive axis remains dormant. On a long summer day, daylight extends far enough that it crosses the threshold, overlapping with the inducible window and triggering the physiological cascade. This detection bypasses the bird's eyes entirely. The light penetrates the skull and is absorbed directly by deep brain photoreceptors located in the hypothalamus. Biological time is measured only when external light physically intercepts an internal rhythmic window. Once that overlap occurs, a neural signal travels from those deep brain photoreceptors straight to the pars tuberalis. The pars tuberalis responds by releasing a molecule called TSHB, sending it toward the brain's third ventricle. This triggers a strict genetic toggle. TSHB downregulates the DIO3 gene and simultaneously upregulates DIO2. That DIO2 surge drives the production of T3, the physiologically active form of thyroid hormone. Down at the median eminence, glial cell end feet form a physical barrier, clamping over axon terminals to block the release of reproductive hormones. The newly generated T3 reaches this barrier and causes these glial end feet to physically shrink and retract. With the physical blockade removed, gonadotropin-releasing hormone, or GnRH, flows freely into the portal blood. The gonads activate and the breeding stage begins. A bird's seasonal readiness is controlled by the physical retraction of a cellular blockade in the brain.
Breeding requires massive caloric expenditure, but so do molting and autumn migration. A bird cannot sustain all three. The reproductive system must shut down to allow the body to redirect energy. To guarantee this shutdown, many species utilize absolute photorefractoriousness. This is a hard-coded cessation of GnRH release that forces the gonads to regress, even while the long, stimulatory days of summer continue. Opportunistic breeders, like the zebrafinch, have evolutionarily lost this absolute shutdown function, allowing for their continuous scheduling. For temperate birds, the on-off switch is rigid, but they do fine-tune the exact timing of the shutdown using non-photic cues, like ambient temperature. This chart tracks great tit testis length across 3 distinct latitudes. While their exact peaks vary, higher temperatures consistently cause a delayed termination of reproduction across all populations. We must ask, is this variation in timing simply localized behavioral plasticity, or is the baseline code genetically hard-coded?
Researchers tested this by comparing European and Siberian stonechats in a common garden experiment. This top graph shows the distinct photoperiod curves these two subspecies typically experience in their wild habitats. When raised in a lab under identical artificial daylight, they maintained their native schedules. The European birds kept their prolonged reproductive window and protracted molt, while the Siberian birds executed a highly compressed cycle. Furthermore, hybrid offspring of these two populations exhibit an intermediate schedule. The timing is inherited. A bird's annual schedule is a rigid neuroendocrine program, genetically adapted and tuned over millennia. Because the end of one stage, like the GnRH shutdown, chemically permits the onset of the next, like molting, these life history stages are sequentially locked. This creates carryover effects. An energetic delay or extra exertion in one season exacts a direct biological penalty on performance in the subsequent season. We can track this cascading penalty in the Manx shearwater. Here is the control pathway, establishing the baseline sequence from chick rearing down to the next year's breeding success. Researchers experimentally extended the chick rearing stage by 16 days, forcing the birds to forage longer and expend more energy before they could transition. If we follow the arrows down the right column, that initial extension delays their fall migration. This leads to a late winter arrival, which ultimately results in reduced reproductive success the following year. Birds cannot view seasons in isolation. An ecological disruption in July is biologically paid for in March. Today, rapid climate change is shifting ambient temperatures worldwide. This causes earlier spring blooms and pushes caterpillar populations to peak weeks ahead of historical averages. This creates a trophic mismatch. Migratory birds are arriving and breeding at their usual times, completely missing these earlier food peaks when they need to feed their young. They failed to shift their schedules because they are locked into an astronomically fixed driver. Day length does not shift with the rising temperature. Even if a species managed to stretch its breeding window to secure more food, the rigid physiological requirements of molting and migrating prevent them from adjusting asymmetrically. They cannot cheat the calendar without triggering severe carryover penalties. Survival in a warming world rests on whether this hard-coded neuroendocrine architecture can evolve fast enough to keep pace with an altering planet.